Good day everyone, welcome to the course on next generation sequencing technologies, data analysis and applications. In the last few classes we have discussed the mapping algorithms and then we have followed that up with some hands on. So, we have introduced a tool called Bowtie2 which actually does mapping, and we installed this tool in our system, and I have showed you how we can build genome index, right. So, there are two steps in using this tool. The first step is building the genome index, which we did using a reference genome, and then we can do the read mapping once we have built that index, and we have also seen how we can actually do this mapping. Okay. So, in today's class we will be discussing how we interpret this data. Okay. So, once we have generated this output file from Bowtie2, how we can actually interpret this data. So, this is the concept that we will be covering in today's class. So, we will interpret the Bowtie2 output, because this is a very complex file, right, and there are a lot of details in that file. Okay. So, these are the keywords that will come across. One is called SAM format, the other is header lines. So, just to summarize what we have discussed so far and what we have done in the last few classes. So, we have set up Bowtie2. We have used this Bowtie2 build function for building reference genome index, and there are certain options that you can give in the command line when you are using this Bowtie2 build. Then we use this Bowtie2 function itself. So, this is the function that we use for mapping. Okay. And with Bowtie2 there are a lot of options. We have mentioned that some of these options could be useful, and we have also discussed concordant and discordant mapping. So, just to remind you again, right, this is how we run this command. Okay. So, there are these inputs that we have to give, right. So, we have this -x, right. So, that actually is the base index, right. So, this is the index that we have built in the earlier step. Then we have -1, -2. If we are dealing with paired n data, we are giving input of these FASTQ files. If we are dealing with unpaired data, we use this -u option. We do not use this interleaved option, right. We do not have that kind of data, and -s is the output file format. So, this is the SAM format. Okay. So, this is something we will discuss towards the end of this class. Okay. So, again there are three types of options that you can give. One is input options, right. So, you can specify what you want to do with the input data. You can ask Bowtie2 to do something with the input data, or you can specify how the data is provided, what is the quality score format, etc. You also have run options when you are running the code, how you should run, what kind of alignment it should do, and then finally output options, right. What kind of output you want, whether you want certain outputs or not, that you can specify using output options. So, what we are going to do, so I have introduced them in the last class or so. So, I will just summarize some of these, because what we will do today is we will also start with using them, right. So, I will show you how you actually use them and what kind of effect you will see on the data analysis. Okay. So, these are the input options, some of the input options that we have mentioned, -5, -3, and then you can also specify this quality score format, right. So, if you are dealing with old Illumina data, you will probably specify FRET 64, otherwise by default it will take FRET 33. In the run options, you have two options, right. So, end to end and local, and inside this end to end and local you can also specify the speed and the accuracy, right. So, there is the speed versus accuracy trade off. You can say you want very sensitive or very accurate result. So, you can say very sensitive, right, or you want very fast, you can also specify very fast. Coming to the output options, you have this -FR, -RF, -FF. We have discussed this in the last class. -FR is useful for Illumina. This is valid for Illumina data when you are dealing with paired n data, right. So, these options mostly apply to paired n data, not single n data. You also have this no mixed option, right. So, this actually gives you or leaves out certain type of alignment, right, which you may not require. You might say we do not require that kind of alignment. So, specifically what no mixed will do is it will leave out this single end alignments, right. So, what happens in paired end alignments, the program will try to map both read 1 and read 2, right. if it does not find any valid concordant or discordant alignment, what it will try to do, it will take these individual reads and will try to align them separately. Okay. So, if you do not want that, because this may not be useful, so we can always specify minus minus no mixed. In some cases, we may not really require minus minus no discordant. For example, we are doing some sort of genotyping, right. We are looking at genetic variation at certain locus or in a few regions of the genome, right. So, we might want to say we do not really need this discordant mapping. So, we can specify minus minus no discordant, right. So, we discussed what is concordant mapping, what is discordant mapping in the last class. And then you can also mention the minimum fragment length and maximum fragment length using these options minus I and minus x. So, we have not used them, right, in the run that we actually did in the last class. Okay. So, what we are going to do is we will actually try some of these variations, because they are useful and you will see how the results will change. Okay. So, let us move on to the terminal, right, and see how these options will change the output. Okay. So, coming to the terminal, right, you remember this from the last class. So, this is where we actually left. We actually ran this Bowtie2, right, using this command Bowtie2. We are calling this program Bowtie2 here. We are giving minus x, right, this is the index file name ist underscore wg, right. We created this in the when we built the reference index using Bowtie2 build function. Then we are giving this input file names, right. These are minus 1, minus 2 means this is paired end data. So, that is why we are giving two input file names. So, one is the iteration 1 read 1 trimmed .fastq, and the minus 2 is the read 2 trimmed fastq, right. So, I mentioned why we need to provide trimmed data, because this is the pre-processed data, right. So, we have done some pre-processing. For example, we have done adapter trimming. So, this is the most appropriate file for the mapping, right. And then followed by that we have this minus s output.sam. This is the output file where the alignment results will be stored. Okay. So, let us try a few options and then we will see how the alignment file actually looks like. As I mentioned, this output file is quite complex. It is a lot of data in there, and we need to understand how this data is organized and what they mean, right. So, if you want to process these SAM files for further analysis, you need to understand this format and the information that is stored inside. Okay. So, let us try different options. So, once you run this, what you get is some sort of summary statistics, right. So, here we have, so what is telling us is that there are 25,000 reads, right, and out of these 25,000, right, so all of them were paired data, right. So, and then there were 2,350 that could not be matched concordantly. So, it was not mapped. There are about 19,500 reads that align concordantly exactly one time, right, and then you have 3,000 reads that align concurrently more than one time, means they are mapping to repeat sequences, right. There are multiple mappings. Then out of those 2,354 that did not align concurrently, right. So, the program then tried doing this discordant mapping, and it found discordant mapping for 1,608 reads, and rest of them, the 746, they were aligned independently, right. So, that is what this minus minus no mixed option, if you give, it will stop this alignment. Okay. So, let us try those first two commands, right, minus minus no mixed and then minus minus no discordant. Okay. So, and see what actually happens, right. So, we can run these commands, right. So, I will run the same command, but now with these options. Okay. So, here I am going to give these options. You can see here, right, my cursor minus minus no mixed. Okay. So, here is this minus minus no mixed, and rest of the options remain same. Okay. So, the options come after the program, right. So, as you have seen now in Linux, we run a program or run a command in the terminal, right. Every time we do that, if we want to give some options, we have to give this after this command. Okay. So, this is what we are doing. So, we can just give this minus minus no mixed and run, right, press enter and we run, and you will see the output statistics, right, and you will see that statistics is bit different, okay, because we have switched off this mixed mapping, means it will not try to align these reads individually. Okay. And immediately you probably notice that this overall alignment now drops from 98% to 92% roughly, right. So, this is an effect that because we have stopped some part of alignment. Okay. So, this is actually closer to the actual mapping percentage, right. This single end data may not be always useful, the individual mapping may not be always useful. Okay. And the rest of the statistics that remains kind of same, right. You have seen something like 25,000 reads, right, 2,354 they did not align concurrently, 1900 738 they aligned concurrently exactly one time, and then 2,900 aligned concurrently greater than one time. So, what is happening, you will see probably slight increase here in these numbers. What it means is that the program is trying to align these concurrently somehow, right. It is reporting some concurrent alignment which it was probably ignoring last time. Okay. So, this will change change the results little bit, okay, not a lot. We can also try this minus no discordant, right. We can simply add this to this command, right. We can have both minus no mixed as well as minus no discordant. Okay. So, let us try that. Okay. And I have just written minus no discordant, and then I will just press enter and it will run again, and it will give you some statistics, right. And what do you expect? You will expect probably the alignment rate will go down further, right, because we are not allowing discordant mapping either, right. So, this is something that you will notice here immediately is that this 90.58% overall alignment rate, right. So, which is lower than 92 and much lower than 98%, right. So, this is the alignment that we get. So, mostly the concordant alignment. Okay. And in many cases, in many applications, maybe we are just interested in this concordant mapping, and we really do not want or need that discordant mapping or mixed mapping, right. So, if that is not required, we can actually stop those, right, and that will make the program a bit faster, right. So, because it will not try all those things. All right. So, if this is clear, right, what we can do now, we can actually try other options. So, these are mostly output options, right, what you want in the output file. Okay. What we now we can do, we can actually also change the run parameters. Okay. So, one of the run parameters that we often use is the local option, right. So, as I have mentioned, it makes a lot of sense to use the local alignment instead of the global or end to end alignment, because in reads, right, especially in Illumina, we have seen the quality score is low in the beginning, and as well as the quality score drops towards the end, right. So, if you are doing local alignment, the program can ignore some of these bases that are of bad quality, and they may not be matching very well with the reference sequence. Okay. So, we will try that now, right. So, we'll come back to that original alignment, right. So, let me clear the screen, okay, and we'll come back to that original command, right, and I'll run it again. And here, so the default is end to end, right, which is the global. So, it will try to match the whole read against the reference sequence, even if the match is a bit poor, right. So, all right. So, this is the result, right. So, you have this 98.26% alignment rate, overall alignment rate. Okay. Now, what we will do, again give this option minus local. Okay. As you can see, I have just given this minus local option, means I am asking the program to run in local alignment only. Okay. And I will run this. What you will notice is that the overall alignment rate will go up. It should go up, because it will be able to now align more number of reads properly to the reference, because it can now remove or maybe ignore certain bases at the end of the read. Okay. So, let's see, right, it will give the results in a moment or so. Yeah. So, you will probably notice this increase in the alignment rate, overall alignment rate, right. So, here are the results. You can see slight increase. Okay. 98.46%. Okay. So, you might say this is not a lot. Okay. And that is the reason is that because we have already trimmed the data quite well, right. So, we have done this pre-processing step, and we have trimmed the data already, and we have seen the quality score is pretty good, right, in the beginning, and in the end they are not really bad for this data. If you had this quality score low quality score at the end or at the beginning, right, we might see a better improvement, more improvement in this alignment rate. Okay. And this will be clear if we now try to run this using the original data where the data is not trimmed. Okay. Because if the data is not trimmed, the program will have hard time for in finding proper alignment for some of the reads, right, where the adapter is still there. Okay. So, what we will do, I will demonstrate, right, how this will change, right. You will see a much more significant improvement if we are doing this alignment or mapping with the original data, right, without any pre-processing, without any trimming. Okay. So, let us do that. So, for doing that, what I will do, I am running this in normal mode, which is the end to end mode, the default mode. So, only thing we need to change are the input files. Okay. So, the name of the input files. So, here sorry, it is D12, right. This is the original file, right, read 1, and then we have the read 2. Okay. So, I have given these original file names now, and we are running this Bowtie2 on these original files. So, let us run and see what the output is. Okay. And of course, what we will expect compared to the earlier scenario, the alignment rate will be much lower, because you have these reads that are not trimmed. So, the program will not be able to find proper alignment. Okay. And immediately you can see this, the alignment rate drops to 77%, right. So, from 98% you have a drop to 77%, because you have these adapters at the end and the program is trying to use them for alignment and it is not finding good match in the reference sequence, right. So, that is why this problem is. Okay. All right. So, what we will do now, let us try this in the local mode and see if there is any difference or if there is any impact on this alignment rate. Okay. So, we will use this option minus local, right, and let us run this and see the output, and there should be a significant improvement. Okay. And that kind of will tell you, right, if your pre-processing step is not very effective, there are some issue, or maybe you may not have noticed certain issues and probably did not take any action, then you will probably see a good improvement in your alignment rate. Okay. So, let us see, right, it is taking a bit of time because it is running in the local mode and you have all the options open, right, the mixed alignment as well as the discordant, and immediately you see there is a huge jump in the alignment rate. Okay. You can see here the alignment rate now is 98.4% compared to the earlier case, right. So, it is very comparable to that, right. So, compared to 77%, you have reached this 98%. This is comparable to the run using the pre-processed data. Okay. So, maybe the statistics are slightly different, etc. This will happen because the program will not run exactly in the same manner, because you have these adapters, etc. But still we now see pretty good alignment rate. Okay. So, this kind of tells you, okay, right, probably it makes sense to run the program in minus local mode, because in case there are some sort of quality issues remaining with the data and you have not done any pre-processing, for those minus local option can still take care of those issues, and it can actually map reads and ignore those quality issues or maybe the contamination issues. Okay. All right. So, I think we can now go back and look at the output file. Okay. So, the output file is the SAM file, output.sam, that has been generated. Okay. And we will start with that file and we will start looking at what you have, what kind of information you have inside those files. Okay. So, I will first go back to the presentation. I will just mention some of the terms, because I do not want to show the file immediately, because you might get a bit confused with all the information in there. Okay. So, let us go back and I will introduce this SAM and BAM format, and then we can come back and see what you have in the actual data. Okay. So, the output file that you have seen is generated in SAM format, okay, and you also sometimes get this in BAM format. Okay. So, the SAM stands for sequence alignment map, okay, and this is a human readable format. So, we can read this, we can open this file and we can read this, which we will do a bit later. Okay. But corresponding to this SAM file, you also have a BAM file, right. You can generate this BAM file from SAM file, and this is a binary alignment map, means the machine can read it, right. So, this is a machine readable format. that we can supply to the machine, but we are not able to read this file. Okay, so if you want to read this file, if you want to write a code to process the alignment results and to identify, for example, genetic variants, etc., you will go with the sequence alignment map. Okay, so the SAM file, and we'll see what are the fields that are there. Okay, so let's discuss the SAM format, what it contains, because this is the file that we will read. And in SAM file, you have two sections. The first section is header section, you will see at the top of the file, and this header section is optional. So, in some cases, in some files, you may not see this header section at all, and you can also remove this header section yourself. Okay. By default, Bowtie2 will generate this header section in the SAM file, but you can switch off this header section. Okay. We will see when you actually come to alignment section, we want to see the results from alignment section, we can switch off the header section. Okay. And the second part is the alignment section, which actually contains the actual alignment or mapping results. Okay. So, the header section kind of describes the reference sequence, what program was used, etc. All these informations are there, the reads, etc. The alignment section contains the actual results for each read. Okay. So, it will have the information where the read mapped, which location, which chromosome, for example. Also, the quality of mapping, whether it found some mismatches, some mutations, some insertions, some deletions, etc. All those informations are there. In addition, it will also show whether you have concordant mapping or discordant mapping if you are working with paired data. So, all sorts of information are there, and we will go over those in the next class. Okay. So, let us look at the header section, what it means, what kind of information it has, and how do you interpret this data. Okay. So, the header section starts with @ sign. Okay. So, it is like a FASTQ format, right. You have this header section that will start with @. We will see in the file, actual file, and you will see that you have this @ sign. Okay. And then you have the SAM format version that is specified using @HD VN tag. Okay. VN means version. So, @HD. So, @HD means header, right, is part of the header, and then you have VN means version. Okay. So, this SAM format, that there is upgrade, right, from some version to another, and then you need to know, right, which version you are working with, because there might be subtle changes here and there, okay, with the information that is present. Okay. So, that is why this version information will be there. Now, you can have multiple header lines, right, not just one, and each header line will also start with @ sign. Okay. So, it will convey some information, but everything should start with this @. Okay, because then if you are writing a program, you can identify, okay, this is the header line, and maybe some information you are looking for, you can also parse or you can gather from the header. Okay. Now, @ sign, then this is followed by a 2-letter code to denote what kind of record you are looking at. Okay, the record type. Okay. So, there are specific notations. So, we say @HD SO, right. So, @HD means header, right, and SO signifies you are using sorting order of alignments. You are looking at how the alignments are sorted in the file or whether they are unsorted. Okay. So, these are the values you will get, the values called unknown or unsorted. So, unsorted is the default option, you will see this in most of the files, and then you can have sorting by query name, right, by the read name, or sorting by the alignment sequence position, right. So, you can have this sorting by reference sequence name and positions, and this order of sorting, this will be defined in another header line, okay, that will start with @SQ. Okay. So, we will come to that, what is this @SQ. Okay. So, you have @HDSO, then you also have @HDGO, means how the alignments are grouped. So, this is again specific, you can be specific, right, you can have grouping by none, right, there is no grouping of alignments. The alignments are grouped by query name, right, where the name of the read. So, the query is the name of the read, and then you can also have grouping by the reference. Okay. So, if let us say some reads are aligning to chromosome 1, you probably want to group them based on the position or the reference genome where they actually align. So, chromosome 1 reads mapping to chromosome 1, they will be grouped together. You can have reads mapping to other chromosomes grouped together as well. Okay. So, this is not necessarily sorted, but you can have this grouping by query name or grouping by reference on the mapping location. Okay, so you can also have the, so beyond the SO tag, right, which is the sorting order, you can also have something called sub sorting order, right. So this is how it will be given, right, sort order colon sub sort. Now what is this sub sorting? So you, if you have sorted these alignments based on something, right, for example, you have sorted based on reference position, right, where they, or the coordinate system, right, where they actually map, you can do a sub sorting after the first sorting using the query name, for example, right. So you can have two levels of sorting, right. So you can have first sorting by coordinate or the position where it aligned, and then followed by the query name. Okay. And the sort order is the same, the sort order value will be same as whatever the value is in the HDSO tag. Okay. Then you have something called @SQ. Okay. So this will give you the alignment sorting order, right, how the alignment is sorted. So that is given in this @SQ. @SQSN, this will be reference sequence name. So star means this information will be there by default, and then you have SQLN star, this is the reference sequence length. So reference sequence name, the genome that you are working with, so this information will be there. You can also have @SQAN, which is the alternative reference sequence name, right. So some reference sequence may have another alternative name that is also given here. You can also have other fields, right, SQAS, TS, SP, right. So you can have genome assembly identifier, right, which assembly that this program is using. There is some description of this reference, and also you can have the species name, right, the organism, right. Then you have something called @RG, which is the read group. This is usually unordered multiple lines, and each line will also have an identifier called read group identifier. Okay, and each RG line should have a unique ID. So these fields you will not see very often, but in case you see, you know how to interpret this, right. Of course, you cannot remember all the details, but you now understand how to interpret these fields if you see them in your SAM file. Okay. Then you have @RGBC. So this is actually barcode sequence that is used by the sequencer to identify the sample, and then you have also some details about the sequencing center name, some details, date of the run, right, programs that were used for processing the read group, right, and you have predicted median insert size, right, the size, etc., and the platform that was used, some sample information, etc., right. Then you have another type of tag that you can get is called @PG. This is a program, right. So which program actually generated this SAM file, right. So this is important, right. So for example, in case of Bowtie2, you should see like Bowtie or Bowtie2 under this value, right. So PG ID means there will be this identifier that will tell you, okay, this is the program that was used. The program name will be there if you have some other name, right. And then you can also have something called @PGPP, right. So this kind of defines what is the previous program that was used to generate this. So you can have kind of define a chain of these programs that have been used on this data before you got this SAM file. Okay. So that's useful, right, information when you are processing the data. You also get this program version, which is called the @PGVM. Okay. And then finally, you can get some comment lines. So they are given by this @CO, right. So these are one line comments, right, and you can have these multiple comment lines describing something or some sort of maybe experimental details or the data processing details, etc. Okay, so let's have a look at the data very briefly, the header part, and you will probably see some of these values that we have just described. Okay. Okay, so let's move to the terminal, right, and we'll see this header part. Okay, so how do you check this header part of this output file? So the output file is output.sam. So let's check the header file. Okay, and then you can use this :C no wrap. So you can remove this wrapping, and you can identify this header part here, right. This will start with this @sign. Okay. So up to this point you have the header. Okay. So that you have @HD, you are using version 1.5 of SAM, right. The first line tells you this VN, right. Then you have SO, this is unsorted, their alignment is not sorted, and then you have the query, and then you have this @SQ. Okay. So this is the reference that is there. So, here you can see some reference numbers. So, these are actually chromosome numbers, right. So, this information is there and length of each of these reference sequences. So, if you check the reference file, the reference file genome data is actually organized in this way. So, that is why the program is reporting these references. Okay. And then finally, you have this @PG which gives you the program, right. So, this is @PG, you have the Bowtie2, right. The program name and ID is Bowtie2 and the program version which is the 2.5.1, and then finally, it gives you the command line that was used to actually get this alignment, get this map. Okay. So, this is how the header line looks like. Beyond this, you have the alignment section which we will describe in the next class. Okay. So, we can quit now and we can go back here, right. So, these are the references that we have used in this class. And finally, just to summarize, so Bowtie2 options can be useful. So, in the first part of this class, we have seen, right, we can use these Bowtie2 options and they can be useful for obtaining better alignment results, and Bowtie2 alignment results are stored in the specific formats, right. So, SAM and BAM, and the SAM is the human readable format, whereas BAM is the machine readable format. And we have also looked into the interpretation of the SAM format header lines. So, there could be some useful information that you might need when you are processing the data. And in the next class, we will be describing the rest of the part of the SAM format files, specifically the alignment section. And thank you.